Our bioinformatics project will be discussing comparative analysis of VP1 proteins across multiple virus families by Grace Wang, Mele Lato, and Ayushi Kathari for General Microbiology in the College of San Mateo.
For our introduction, the purpose for our topic is to explore how viruses with VP1 proteins compare across different viral families. Our focus viruses are norovirus, Coxsackie virus, A20, A21, and B3, infectious bursal disease virus, or IBDV. The norovirus is from human gastrointestinal virus, major cause of stomach flu. Coxsackie virus is an enterovirus infecting respiratory and GI tissue, causes hand-foot-mouth disease. The IBDV, or the infectious bursal disease virus, is a bird-only infection. VP1 functions as RNA-dependent RNA polymerase, not a capsid protein. VP1 importance are the structural capsid protein and helps with host attachment and immune recognition. Our big question is, do viruses with similar tissue targets, for example, gut or mucosal tissues, share VP1 similarities, even if they are from different families? The bioinformatics allows sequence alignment, evolutionary comparisons, and identification of conserved regions. For our objectives and goals, they are to explore whether viruses found in similar environments share genetic similarities in their VP1 protein, particularly in norovirus and related viruses. Also, compare VP1 amino acid sequences using bioinformatics tools such as online databases, sequence alignment, and phylogenetic analysis. Another is determine whether evolutionary clustering reflects shared tissue tropism or similar host entry mechanisms among different viral families. Our hypothesis is if viruses infect similar tissues or use similar entry mechanisms, then their VP1 proteins will be more closely related evolutionarily.
Materials. For this project, we used a computer with internet access to perform all analyses. We accessed the NCBI protein database to download FASTA VP1 amino acid sequences for our viruses of interest. We used genome.jp as the interface to run clustalW, which allowed us to align all the VP1 protein sequences and identify conserved regions and similarities between the viruses. Finally, phylogeny.fr was used to generate phylogenetic trees to visualize evolutionary relationships. And the sequences downloaded consisted of 6 VP1 proteins, consisting of norovirus, Coxsackie virus strains, and infectious bursal disease, which allowed us to compare viruses infecting similar tissues.
So our first step was to conduct some primary research on the norovirus because we decided that would be our primary focus. And when we did that research, we found that the VP1 capsid protein was really important to the norovirus, and we identified that as our mechanism of interest. So following that, we identified other VP1 containing viruses, and we downloaded their VP1 FASTA sequences from the NCBI protein database to analyze. We then performed multiple sequence alignment using genome.jp with those FASTA sequences to basically compare their similarity scores and see how similar the protein sequences for these different viruses, and specifically their VP1 spike proteins, were. We collected those pairwise similarity scores for comparison and analysis, which we'll see on a future slide. Then we also uploaded our FASTA sequences into the phylogeny.fr website to generate phylogenetic trees, which we then interpreted their clustering patterns to identify evolutionary patterns and, again, similarity between the VP1 capsids of the viruses. So on this slide, we can see two different versions of the phylogenetic tree that was generated by phylogeny.fr using our 6 viral sequences. On the left, we have a radial phylogenetic tree, and on the right, we have a linear branching phylogenetic tree. In both trees, they demonstrate the exact same information. So in both trees, we can see that the viruses are first and foremost grouped by the virus family they belong to. So the Coxsackie virus, the 3 samples for that are very close to each other, and the norovirus, 2 samples are very close to each other, with the IBDV pretty separate. So what we can see from this is that even though we can point to maybe different strands of the Coxsackie virus that are more similar to the norovirus or more similar to the IBDV, the spike proteins for these sequences really do group based on their virus family. That does seem to be a significant factor, and very less so the tissue they infect. Now, as we can see on this slide, the output from Genome.jp, which we ran pairwise correlation scores between all the FASTA sequences, that data also supports our phylogenetic tree analysis. So we can see on this slide that we ran pairwise comparisons between all 6 sequences, and the sequences that were the most aligned were sequences 1 and 4, which are both Coxsackie virus, sequences 1 and 6, also both Coxsackie virus, sequences 2 and 5, which are the 2 norovirus, and sequences 4 and 6, both Coxsackie virus. So this really supports our analysis from the previous slide that the viruses and their spike proteins are most similarly grouped based on their virus family. As we can see through the rest of it, there really doesn't seem to be much similarity between any of the other viral spike proteins, all of them hovering around 10 or 11% similarity, even with the Coxsackie virus and the norovirus, which theoretically infect the same tissue, so should have more similar spike proteins. We're really not seeing that here.
Moving on to the discussion, our Clustal W alignments and phylogenetic analysis supported our hypothesis that viruses infecting similar tissues often exhibit greater VP1 similarity. Coxsackie virus A20, A21, and B3 were the most closely related, with pairwise alignment scores of approximately 58, 42, and 38. These results are consistent with the clustering observed in the phylogenetic tree presented in the results section previously. Similarly, norovirus strains showed moderate similarity to each other with an alignment score of about 41, reflecting VP1 conservation within the norovirus lineage. This indicates strong evolutionary relatedness between the respective viral families. However, there was a very low similarity score of about 11 to 12 between norovirus and Coxsackie virus, despite both targeting the gastrointestinal tract. This suggests that the viral family, not just the infection site, strongly influences capsid protein evolution and sequence conservation. Infectious bursal disease virus exhibited low similarity as expected with all human infecting viruses, highlighting the influence of host range and evolutionary history as IBDV exclusively infects birds. Overall, these findings highlight how bioinformatics tools can reveal genetic relationships that aren't obvious from symptoms or tissue tropism alone. Through these tools, VP1 conservation patterns allow us to predict evolutionary history, potential cross-species infection, and guide vaccine development. In terms of limitations and future directions, our analysis was limited by the small number of sequences, which reduces the strength of evolutionary inference. Future research could include additional VP1 sequences such as enterovirus 71, rotavirus, or sapovirus, and examine other genomic regions to improve phylogenetic accuracy. We also face technical challenges including database troubleshooting, differences in amino acid sequence lengths, and limitations in uploading sequences to analysis platforms. Despite these limitations, the project illustrates the practical application of molecular sequence data in microbiology and public health, emphasizing the value of computational tools in studying viral evolution. And here are the references we used for this project. Thank you so much for listening.